Hello, everybody. So I know it's flipped, but I'm going to be going over exam review for our first exam. So right now I don't have anybody in the room, but I'm writing out some topics right now. So I'm gonna go ahead and finish writing those out, and then I'll go over them if nobody kind of shows up. Um, and I'll just do a little general spiel. If anything I say like catches your attention, say like I don't really understand it, just either send a text message on Remind, or you can email me and say like, "Hey, can you make a more in-depth explanation to this topic?" And I can do that for you. Or if you're saying like, "Well, how in depth do I need to know this topic?" I can provide both of those clarities for you. So just one moment, I'll go ahead and finish writing all this out, and I will be right back. Alrighty. So I just finished writing out a few more things. This is all based, oh God, I hope you can see this. It might just be a whole lot of me talking and you looking at words backwards, unfortunately. Um, so, um, just a few things. So there is going to be a lab and lecture section to each of the exams that you'll ever take in micro. Um, although the lecture stuff might tie in, if she lectures on something in lab, it's most likely going to actually be on the lab portion rather than the lecture. So I just wanted to go over a few key points for the lab section at least. I know that for whatever reason, it was a lot easier for me to do the lab section of the lecture. So if you feel like you might excel at that, then definitely devote more of your time to lecture, because in the grand scheme of things, you spend a total of 6 hours every week focusing on lab, and that's only in the lab time. You still have to go home and finish the actual lab, or correspond with your peers, do things like that. So you spend a lot more time on lab rather than the measly 3 hours a week that you would on lecture. So it's easy to fall by the wayside. I had to kind of pick up the slack when I was in the class. I didn't do so hard on my first exam, I'm not gonna lie. So I said, I really gotta get it together and figure out what it is I'm not doing right. And I was devoting a little too much time to the lab and not so much to the lecture. So here we go. First up, lab one. So lab one is all about the charts. Now for the charts, you need to be able to read a chart and say, in this year, this is what was happening, or this percentage of the world is like this, or in Africa at this time, there was this many populated, vaccinated, whatever it might be. I don't personally know the contents of the exam, so I'm just telling you what I know in terms of what I've personally taken and from what Dr. B has told me, because she does correspond with me in terms of drive this, they don't really understand it, or I'm getting a lot of questions about it. So definitely be able to read the graphs and then understand the dependent versus independent variable. I always like to say in my head that the independent variable you don't really manipulate, but the dependent variable depends. That is the one that it can vary. So when you're measuring something, right, let's say weight, for example, if you're measuring weight, that depends on what you're measuring, when you're measuring it, at what point. That would be the dependent variable. That's not a very great example, but just think it depends. I don't know what it's going to be. There's no way to know that, but you do know what environmental settings you manipulate, and that would be your independent variable. So that pretty much covers it for lab one. The charts are probably the thing that I shouldn't have made that stupid mistake. I switched it up. Slowing down and paying attention is going to be a big thing. You have 3 hours for this exam. And I'm going to tell you, a lot of people who didn't score too high finished within the first hour and thought that they just did so well. So take your time, slow down. If you're stressing out, take a deep breath. You have the time. Just keep moving forward. That's the most important thing. So that does it for lab one. Lab two is all about microscopes. You have to be able to look at a microscope and be able to read what's on it. So that includes, the arrangements of the bacteria. So is it a cocci? Is it a bacillus? What arrangement is it in? Is it in long pearly chains like a streptococcus or a streptobacillus, or is it in clusters like a staphylococcus? Is it single? Is it diplo? You have to be able to read those things. Not only that, but you also have to know some of the parts. So it won't be as simple as tell me what this is, and it points to the ocular lenses. It has to be, a little bit more critically thinking, what impacts the resolution of your image. And you could say, well, that could be the ocular lenses, right? Because they have a certain magnification. You could say that the resolution also depends on your stage height. So with your fine and coarse adjustment knobs, you could say that the objective lenses themselves, they impact how well you can see something. The rheostat. Now, if you're up too high in power, if you're all the way to oil and you're still really low on the light, that can affect your resolution, right? So just it's going to be a little bit more critically thinking about those types of things, like what do they do, how they work, how they impact what the image that you're seeing. So that'll be it for the parts. And then also wavelength. Wavelength is another one. This goes hand in hand with like the phase contrast microscope. You kind of have to know what's happening with the wavelengths of the light. But with the wavelengths in a microscope, it's going to affect what colors you see, how you see it, things like that. That's why with a phase contrast, it's like a lot of shades of gray rather than just more black and white. So understanding that and which light rays have more or less energy. Let's see. So oil immersion, also how the light refracts. Why is the oil important? Ask yourself those questions. If you can answer those things, then you're on the right track. One last, what was I going to say about that? Oil immersion, yeah. Oh, how a specimen takes light. So there's a difference between reflection or refraction and fluorescence. So that's how we can see bioluminescence, is that the light refracts off of whatever is bioluminescent and gives us a different wavelength of light. That's why you shine a blue light onto a scorpion and you get like almost a bright white light back. They're fluorescing and returning that light with a different wavelength. Same thing with the jellyfish, right? So understanding how that type of wavelength works as well, how a specimen can affect that. That also ties in with the phase contrast, because remember, when a specimen is hit with the phase contrast microscope, it does shift a little bit of the light waves. So when you have the background being unshifted, you have to adjust for that. So that's basically it for lab 2. Just get really, really comfortable reading microscopes and telling apart which ones. And if you have any questions about those, let me know. And I can kind of make a more in-depth video about that, or I can talk to you between now and the exam. But back to lab 3. So lab 3 is mostly just about your actual lab technique, and that's what you're going to be mainly judged on. But you will be able to, or you will have to be able to understand the difference between a pure culture and a mixed culture. Obviously, you have to also know how to see best, but if you are still uncomfortable with your streaking technique, please come to me. I will help you as much as I can. I know that I struggled, and I've helped those who have struggled before, and they, not to brag, but they have gotten much better technique after a little bit more one-on-one help. And it's no shame. I mean, I didn't know what I was doing at first either. I just kind of guessed and checked as I went. But that is it for lab 3 off the top of my head. Lab 4, the gram stain. So you'll need to know how to actually do the gram stain. Do it well, do it correctly, because you're gonna have to do it on the exam. If you're still uncomfortable with it, again, come let me know. Come let Dr. B know. Maybe we can set apart some time, maybe her office hours, where you can practice maybe a couple more times. So that's going to be part of your lab section. And then you'll also be asked a couple of other things. So you'll be asked questions about the other stains that were presented to you in class. So that'll be like the Schaefer-Fulton test. That'll be like the Knievel test that are, yeah, I keep on calling it Knievel because she calls it Evil Knievel. But I'm sure if you say that on the exam, she might understand. So the Meneval, that's it, Meneval one. But understanding all those stains, how they work, what's the advantage, and what you can see is going to be the most important part about that. Let's see. Oh, yes. What the advantages and the disadvantages are of all the stains. So, like with the gram stain, it's pretty well known. We define most bacteria by gram positive, gram negative. There's only a select few that fall into that, like gray area of they're like neither, or they're like not really a true gram positive or gram negative. That would include like things like syphilis, tuberculosis, those waxy cell wall, like leprae, Mycobacterium leprae. Those are the ones that fall into the gray area. They're not true gram positives or gram negatives, but they might stain a certain way. So understanding pros and cons of all the stains will help you in the long run. Also understanding probably some common bacteria that stain that way. So like the Nielsen one, the Xyl Nielsen one. I don't know how to pronounce it, but I know how to spell it at least. That one is going to help you with tuberculosis. This is going to be the waxy cell wall ones. It's also going to help you with the leprae, the Mycobacterium leprae. There you go. So those will help you detect the waxy cell walls that might not have a true gram stain, or it might give you a false identification. So, what else is there? The Meneville stain, that one's going to help you with endospores. And you might want to know that B. cereus is the type to have an endospore. That was in a case study. So really going back and like from this point forward, understand that the case studies are supposed to be a supplemental way to teach you more information. Those case studies and other projects that you're doing, they're supposed to give you another layer of understanding. So I'm not going to call out anybody, but when I was in class and I was watching a few people present, I did see some people just on their phones. And I was like, come on, but like I would want somebody who's listening to me to give me their full attention. And I also don't think that it's very encouraging. If I was up there nervous and somebody was on their phone, not like wanting to listen to me, I guess, like that would be discouraging. And I'm sure that I don't think that you would want it the other way around. So, can't name any names, but paying attention to those case studies is going to be integral to your education and furthering your understanding in lab. Because I mean, there were a lot of different stains used in those case studies, and they were also supposed to help prep you for kind of how it's going to be like in the nursing world. You're going to be listed with a bunch of facts about this patient, and you're going to have to be like, okay, critically think about all of them very quickly, and then say, okay, this is what we need to do. This test will tell me what I need. And then after this test, I'll be able to make a determination. It's called like a nursing diagnosis, which is, my sister is actually an RN, and she says that they're useless essentially because the doctor just walks in, says what you were going to say, and then leaves. But it is still good to know, nevertheless. Sometimes you can sway an easy doctor. So, yeah. Nurses are always the workhorses of medicine. And they will, 10 times out of 10, probably know better than the doctor. They went to med school, I understand, but they also sometimes have terrible bedside. So, please remember that. Anyways, this lab number 4 is going to be a lot about critical thinking through about like, if you were to forget this step, if you were to overdo this step, if you were to leave this on for less time or more time, what would happen? And walking through those is important. And if you need help, I can also ask, or I can also walk you through that. And I would want to say we could actually like do a failed experiment in the lab, but I don't know if we have the resources or time for it. But if you're curious, I could probably do it sometime in the near future. So moving on to lab number 5, I actually made a really good PowerPoint for this. I'll probably upload it just so you guys have something to use at home. I'll be probably uploading a bunch of stuff tonight. So you really want to know your Msa Emv Blood Auger, being able to read all of those, what makes it selective, what makes it differential. And then you also want to know your broths and F tubes, how to read those, and what makes those unique. How, in what way would you be able to identify those, and how would you be able to determine certain qualities about a bacteria? For example, with the broths, if there was a pellicle, you would be able to know that this is an aerobic bacteria, right? Being able to do that with all of these things would be amazing. Being able to do that on the exam is going to be very important. Lab number 6, Berkey's keys. Well, this lab isn't really covered too much in the actual lab practicum. It's also, in my opinion, a little bit easy. You just kind of have to follow the flow chart. If it is confusing, let me know, as usual. But the gist is that when you're presented with, like, you may know some information. Let's say I have a gram-negative rod. I know that at least. You're going to find that on Berkey's keys, and then it's going to have an arrow. And usually that arrow will say oxidase positive, oxidase negative, some kind of test in positive or negative. So then you have to look at that and say, okay, so is my bacteria positive or negative? So after you determine that, then you're able to follow the flow chart. Then it's going to be another crossroads, right? So that's what Berkey's keys is really all about. You're going to be using it in the future, I promise. It was my best friend. You're going to be using it in future labs. Keep it on you because it's also just interesting, in my opinion. Lab number 7, microbial metabolism. Try to say that 7 times fast. So, with microbial metabolism, you have to be able to tell the different fermentation pathways, understanding glycolysis, understanding Krebs cycle, understanding aerobic versus anaerobic respiration and what those entail, how much energy, exactly how much ATP is produced. I'm going to be honest, I did not study that at all for the first exam. I put it out on a whim. I just wrote down, because I recognized NADH, NAD, FADH, like things like that. I wrote that down, guess, and I put numbers in front of it, and I somehow got like partial credit. Don't do that. Just memorize it. You'll need to know it in the future. So it's a very useful thing. Please don't be like me. But that is going to be the gist of the lab side of things. So in that, I also wanted to draw your attention to, I think I have it almost pulled up. Hold on. Here we go. All right. So this is going to be your Canvas shell, right? I'm underneath exam 1 content. If you scroll all the way to the bottom of this grid here, you'll be able to see the practical study guide 1, and she just uploaded exam 1 review session. So this is, the PowerPoint is going to be really helpful for you in a very broad term, but if you want to know how well you'll be able to do on the lab portion, or if you want to feel like 100% confident in it, review this practical study guide. It's going to say, be able to do this. It might ask you a couple questions, write everything down, answer it as if it was like an actual study guide you had to turn in. That was my number 1 helper. And for that, let's see, first exam, second exam, I missed 1 on the lab practical and I used that. Third exam, I missed none. And I used the practical study guide. And they correspond with every exam. On the fourth exam, it was hard to tell. We actually switched online for a little bit, so um, but I still used it. I still got a good grade. Trust me, I can tell you how to get a good grade in this class. Um, so that is going to be my 2 cents about the lab section of things. Now I'm going to switch gears and we're going to talk more about lectures slash theory behind something. So I'm going to go ahead and erase. Whoop. I'm going to go ahead and erase some of this, just because you can't see it, but this is only like a third of the board that I wrote on. Everything else is filled. So we got a little bit to go through. Right up. I just like lists and checking things off, or whiteboards and erasing them as I go. All right. Whoop. So again, it's probably backwards, but I might not the desk too. There goes the pencil. Anyways, so yeah, this is my little like center exam one review. So there are a few things that we needed to know in terms of the famous people in the 1800s that could have died during childbirth but didn't. So we have immunology by Pasteur, Jenner, and Fleming. So Fleming, a little less immunology and a little bit more antibiotics. So Pasteur, oh, our good friend Pasteur, he has done a lot for us. He was in helping with fermentation and spontaneous generation. He proved spontaneous generation wrong. He helped determine what was wrong with the wine industry and why all the wine was tasting so terrible. But he also did help the immunology side of things. So I'm going to go ahead and pull up the review PowerPoint. I'm just going to hit a few of the really, really strong stuff that you'll need to know that a lot of people kind of bypass. Sorry, it's at the very end. Yes, it's 142 slides. Don't get overwhelmed. You should be already knowing some of this stuff. It should just be like a reminder. Here we go. So where did you go? Okay, here we go. So Edward Jenner was a freak. He decided to take I believe it was like cowpox is what it was called. He decided to take that and rub the pustule and into not even his child, but the milkmaid's child. Rubbed it in there, and then all of a sudden he didn't get smallpox, which is a relative of the cowpox. And so all of a sudden he saw, oh wow, I just gave this kid antibodies and he's now immune. So that's how he was able to contribute, if you want to call it that, to immunology. That's kind of our first breakthrough with, oh, we could do something artificially to gain immunity. So that was Jenner. Louis Pasteur did a little bit something better. He decided to develop vaccines rather than just stick pus into some kid. So he developed the cholera and rabies. So that's rabies. Sorry, my handwriting sucks, but I'm sharing the screen so you guys can at least see it. So those were those two. The other guy is Fleming. So Fleming. So when you hear chemotherapy, don't think cancer. Although chemotherapy is used for cancer, chemotherapy is just treating something with chemicals. So if you've ever taken an antibiotic before, you've undergone chemotherapy. Congratulations. If you want to put that on your wall, but it's used in a different sense nowadays. Chemotherapy in a scientific term is different. So Alexander Fleming used penicillin first. He discovered penicillin, and that was the antibiotic of choice. Antibiotic, penicillin is the most commonly used antibiotic. Um, if you didn't know this, those that are allergic to penicillin are kind of like screwed half the time because that's just how they treat a lot of things. Um, Penicillin is like, okay, this is like the most docile thing we can give you. And it's hard if you want to know, it targets the cell wall. Um, and it is usually a little bit more effective in I want to say gram negatives. Yes, gram negatives. Um, but you won't really need to know that. I just know that he developed penicillin. Wow. Okay, my handwriting sucks. So he developed penicillin. You don't need to know the year, just know it was him. That covers immunology for the most part. I'm gonna switch over because it's right here. I kind of ran out of room and started going back and forth. Um, I'm gonna cover some gram-positive versus gram-negative bacterial wall components here. So the peptidoglycan layer is made up of your NAGNAM cross-links with the tetrapeptide bond links. So understand that you just need to know the NAGNAM and you just need to know the tetrapeptide. You don't need to know like if it's L-lysine or something like that. All you need to know is NAGNAM and then the peptides. Okay, that's about it for the peptidoglycan. Also understanding, the difference between gram-positive and gram-negative will help you a lot. One thing about gram-negatives, though, they have that outer membrane, right? That outer membrane does contain a LPS, lipopolysaccharide, and that lipopolysaccharide has a lipid A portion, which is an endotoxin. Okay. Is it endotoxin or exotoxin? That'd be a good thing to understand. I just don't want to tell you guys the wrong thing. Is it all the way at the bottom? Oh, I swear I'm more organized. Okay, here we go. It's an endotoxin. Okay, I was right. It's an endotoxin. The reason why it's an endotoxin, though, is because an endotoxin is released once it's broken. An exotoxin can be released while the bacteria is alive. So this gram-negative bacteria has to be lysed in order for that LPS and lipid A portion to be endotoxized, if you want to call it that. So understanding that what section makes it toxic, et cetera, is going to help you out a lot. Keep on saying that phrase. I need to get a new one. Anyways, erase this. I definitely encourage you to review those slides and once you review it, make a list of things that you know and like 100%, like you've got this. You can ask me once you like first thing in the morning, as soon as you wake me up, if you ask me a question about, the cell wall components, I will be able to answer that no problem. Like, and then if you're like, well, maybe it'll come to me, but it'll take a minute. Like those types of questions, highlight is like a maybe, and then kind of keep going from there. I'm going to sit down now because I'm suddenly tired. All right. But I got a swivel chair, so I can still move. All right. So next step is going to be the endosymbiotic theory. Now, if you don't know what this is, as soon as it comes to mind, maybe study it. So the endosymbiotic theory is going to be the theory that at one point, there was a bacteria living its life, hanging out. Here, I'll draw it out. Why not? There was this little bacteria living its life, hanging out. Who knows if it had flagella, but let's say it did. That'd be a fun little thing to have. But all of a sudden, dun dun dun, this another bacteria came by. And it was just a little small bacteria. So it can make energy too. So he was like, can I just live in you for whatever reason? And they joined forces And this is technically a parasitic relationship where this one is going to be now codependent off of this one and vice versa. So once this guy came in, we were one. And what did this become, you might say? Because there's always remnants of things from long ago, right? I mean, just look at like, what is it? There's certain bones in like a walrus or something that they don't need anymore, but they're evolutionary. There's always a trace. So this became the mitochondria, and this became the eukaryote. Oh, let's write it like that. The eukaryote. So that's what the whole of the endosymbiotic theory is. It's how eukaryotes came to be. All right. And of course, if you need any more help, let me know. But that's the gist of it. That's about as much as you'll need to know. I don't think you'll need to know who developed it, just really what it is. All right. Next up is going to be enzymes. Favorite. Enzymes. So be able to draw this from memory. And I promise you it'll save you so much time. So we have our inactive enzyme, right? So normally there is something that binds here, right? And it looks exactly like this. So we have our, let me find the exact verbiage so I don't confuse anybody. Oop, 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 oop. Okay. So I have my enzyme and then I have my substrate. That's the word I forgot. Now, as we all know, life likes to be difficult. So sometimes the substrate can't bind and it cannot undergo the process it wanted to. And that's because of a couple of reasons. One might be this competitive inhibitor decided to just hang out and alter what the binding site looked like. And because of that, the normal substrate couldn't bind it. No more enzymatic activity, nothing. Now, this can be permanent or it can be reversible. Remember that. So, that's one way. Another way, let me just erase this one. Another way is that was a competitive inhibitor, right? Now we have a non-competitive inhibitor. That's just this little guy right here. He'll bind on here. And he'll say, whoa, 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 what's going on? I don't feel so good. And it'll all of a sudden make this look different. Now this can't bind here, no siree. So, no enzymatic activity. There are a few things that can alter the ability for even a normal enzyme to undergo enzymatic activity. That would be included here. Temperature, pH, enzyme, and substrate concentrations. So too much substrate and not enough enzymes means that there's only so much enzymatic activity going on, and too much enzyme and not enough substrate means, whoa, 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 we're really in the depths here. Why isn't our substrate being produced? And then of course inhibitors, which we just talked about. You guys should be able to know about proteins. I feel like this was talked a lot about in A&P, chemistry even. Know this one. So remember, enzymes are made of proteins. So at a certain point, if it gets too hot, which we were talking about the temperature, right? If it gets too hot, you denature the enzyme. Now, there's only a couple of enzymatic, there's only a couple of proteins that can be undenatured, if you will. So renatured, I guess. The most common one is milk. So if you were to heat milk up, the proteins in milk can be reversed and cool back down. So it can go back to its normal state even after denaturing those proteins. But most other things, like think about cooking an egg, those proteins cannot be reversed. So understand that about 37 degrees, which is, at what temperature we cook our bacteria, is optimal for enzymatic activity. And remember, enzymatic activity isn't just like this once in a lifetime thing. We're always undergoing enzymatic activity. That's how we digest our food. That's how the bacteria grow and digest their food and create a more apt environment for themselves. Let's see. There you go. Substrate. Now, sulfonamide. I know that this one is like, ooh, big sciencey word, pharmacology, maybe. I have no idea. I understand that it looks scary, but it's not as scary as you think. So, usually, a sulfonamide will bind with PABA and create folic acid, but a competitive inhibitor just takes up the whole binding site, and instead of sulfonamide binding with PABA to produce folic acid, sulfonamide binds with sulfa. It gets confusing, but sulfa, and there's no folic acid produced. But yes, this is just an example. Think of it like an example. Don't think of it as like some other scary like chemical biological thing that's going on. Although it is those things, it's not as scary as you think. I want to say that's okay for now with enzymes. Yeah, enzymes are just always important. And if you took A&P already, A&P at least 32, you should have already known a little bit about enzymes. But yes. Did I erase? Oh, dang it. Yeah, I erased one. Oopsies. All right, let's move on to our kingdoms. Can you see that? I'm gonna put my hand. Okay, yeah, you can see all those. So on the prokaryote side, we have our bacteria, we have our archaea, and on our eukaryotic side, we have our plantae, we have our animaliae, we have our look behind me. Protists and fungi. Haha, I got them all. Yay. Now repeat those to yourself until they are drilled into your brain. Okay, here we go. So, main difference between these two guys, archaea does not have peptidoglycan most of the time. I'm going to say most of the time because there's always an exception, and that is a rule in biology. That is the only definite thing in biology, is that there's always an exception. Biology is just crazy. But we study bacteria. We study the peptidoglycan and some of the bacteria that lack the cellulose. But the archaea, no peptidoglycan, always lack cellulose, almost always. Big defining characteristic of prokaryotes, no nucleus. However, on the eukaryotic side, everybody has a nucleus. Woo! You get a nucleus. You get a nucleus. Anyways, I'm pretty sure that you might be able to make some examples of protists and fungi, plants, and animals. Right? We're animals. Plants are like the houseplant you have. Fungi, that could be, you need to know this for sure. Unicellular yeast or multicellular mold. Those are going to be really important under fungi because he's a fungi. Protists. Protists are like, just think of like a bog. Like you have no idea what's in there. Algae. You might think that algae is actually a fungi, but it's not. Because what they do is they like, I want to see if there's a picture in here. There's no picture, but they can bind together. They stick to each other. They're just little sticky things. Binary fission for DNA, or for bacteria. What the heck? I mean, they have to copy their DNA, but yes. Oh, archaea. Okay, so archaea, they live in the extremes. If you see the picture on the bottom, that is I forget what exactly. I think it's called the where's that? Oh, something like that. I want to say miasma, but I know it's not miasma. But it's in Yellowstone. I went to Yellowstone not too long ago. First of all, it's beautiful. Second of all, people are stupid. Currently, there are people walking around that part of Yellowstone and they're getting boiled alive. Yeah, you heard me, boiled alive, because they don't understand that if you look at that picture, that body of water is constantly boiling. It is constantly hot enough to do that. It's also a very salty environment. If you look at the ground, everywhere, it doesn't matter where you are, it's always salty. And also because of the water vapor constantly coming off and then cooling back down in the atmospheric temperature, everything's wet all the time. So that area is a perfect place for archaea. Yay. So, and also hot springs is another place. I've only been to a hot spring once, but I didn't get a brain-eating archaea or anything in my blood. So that's a pro. Moving on. But yes, know that bacteria will find a way. They will always grow. They will always be in the weirdest nooks and crannies. But archaea are freaks and they like it that way. They like living on the edge, living like Larry. Alrighty. Fun guys. Yeah. So remember, fungi are where we get our penicillin. Penicillin is a fungi. Penicillium is its fungal name, right? Unicellular yeast, multicellular molds. Let's see. Oh, so I'm going to talk about this more later, but where they derive their energy from, the big thing. They are saphites. Man, why is the word slipping me now? They eat dead things. That's what their thing is. Man, I wonder if I can find it now. It's gonna bother me. Give me just 1 second. I swear. Oh. Hold on. Also, a great little note is to look in this area. Chemoheterotrophs, yes, but they have a sapphire, sapphire, something like that. I'm honestly so bothered by it. I'm gonna Google it, and you guys are gonna join me. Okay, we're back. Saphites. Sapphire, no. Um, Sapphitic. Sapphirites. Okay, I'm gonna okay, well I'm saying it, but I'm saying it wrong. It's like the words in my head, but I'm okay. Moving on. Because that's hard. Also know that their cell wall is mostly made of chitin. She loves chitin, let me tell you. I did not know that on the first exam, and I was like, this fungi have chitin, I swear. Like literally the rest of the year. Anyways, eukaryotes. They have the true nucleus and they can move. They have tons of different ways they can move. So protozoas. They're the ones who have the pseudopods, cilia, flagella. Usually they're also kind of funky. They really do anything they want to. What else we got? If you when in high school, you always learn about plants versus animals. It's not that important to cover because if you ever think about human anatomy and physiology and biology, you're going to be able to understand that, yes, we're different from plants. Yes, we get our food from outside sources. No, we don't undergo photosynthesis. And understanding that our cellular respiration is the exact opposite of photosynthesis. So that is going to be your kingdoms. Review it, know it, test yourself, make Quizlets, whatever you need to do in order to understand 100%. I'm going to go ahead and erase that. I know I'm obnoxious right now. And then we're going to move over to the other half. Yippee. These are just a few of the important ones that I feel like everybody misses. So I'm making sure to go over it. People get intimidated by antibiotics. Yes, antibiotics. I have them right here. They're obviously backwards, and I feel bad for it, but antibiotics, know for a fact erythromycin and chloramphenicol bind to the 50S portion of the ribosome. You don't need to know exactly what that does yet, but you will in the next few weeks. But knowing it now helps. Streptomycin and gentamicin, they're going to bind to the 30S protein part. You'll also know what that means later. But just know the different numbers, and at least you'll have these memorized by the time we go to antibiotics. It's gonna be a lot of antibiotics to memorize. One big thing is nomenclature. So nomenclature, remember that the genus is going to always be capitalized. So we have Bacillus cereus, Staphylococcus, Staphylococcus. Wait, what is Aeruginosa? I don't know that one, and I can't pronounce that one. Uh-oh. Well, anyways, the genus is always capitalized, it always comes first, and you always underline or italicize. The epithet comes second. The epithet, man, I'm sounding like Elmer Fudd, just stuttering all the way here. Those are gonna be your more commonly used, or like most commonly spelled all the way out. Okay. Moving on to some spontaneous generation. So spontaneous generation is the idea that life can come from nothing. Now, remember, we weren't as smart as we are now a long time ago. And not to speak for the entire scientific community, but also for our career education. So thinking about spontaneous generation, you have to say, okay, who liked it, who didn't? So you'll have to know, Reddy, he's the one who did the spoiled meat. He did the 3 different jars. One had a mesh cloth, one had I think a cork on it, and one was just open. And you could see how those different ones generated maggots. That was his whole spiel. Pasteur, however, oh, how we love Pasteur. He did the Swan flask experiment. The Swan flask experiment was kind of revolutionary because he kind of proved it that life has to come from somewhere. And I mean, think about how many discoveries this guy had. He's had a pretty good standing with the scientific community. I think that he kind of had the final say with, I performed this experiment. I'm right. I'm pretty sure that's how it went. So remember, spontaneous generation is the idea that life can come from nothing. And the idea of biogenesis is the idea that life has to come from somewhere. So the idea is spontaneous generation says the the fly eggs can just appear, while biogenesis says no, the fly has to lay them first. So yeah, that will be that. In the same vein of some old people that discovered stuff, Koch's postulates. Now, Koch did that really unethical experiment with the bunny, the several bunnies. So the idea is that if you have a bunny and it dies for whatever reason because of a bacterial infection, we can isolate that from patient zero. Patient zero died because of this. We can put it into a pure substance. And we inject that into 3 other bunnies, unsusceptible, and one of them dies. The idea is that we have to be able to isolate that same bacteria from the one that died, because that means that is the causative agent. Once we isolate it, we know it's the well, the 4th rule is that causative agent has to be in every single case. So you can't say, oh, well, this disease, let's call it, I'm in a math classroom right now, and all I can really think about is naming a disease math. So let's say you have a disease named math, and you say, okay, well, this person died because of math. Well, did you isolate that causative agent, math? M serious, I'll call it mathematics serious. Did you isolate that from this patient? And they say no. Well, then this isn't math. That's kind of what we have to compare it to. But yeah, review Koch's postulates. It's only the 4 rules. But moving on. Fermentation. Pasteur was the big guy on it. Understanding all the different pathways and what exactly they produce is gonna help you a lot. I'm gonna fix that. Energy, glycolysis. I kind of already said this before, but no, the Krebs cycle and the electron transfer chain. What exactly is used? What exactly is spent? What exactly is produced? What do you get at the very end? How much energy are you making net and at every stage? That is going to be a lot. I need to stop saying that. I'm sure it's getting annoying. I'm sorry. One last thing is, I said this in class earlier today, but oil rig. So oil rig is the nice little acronym to help you remember oxidation and reduction. So oxidation is a loss, oil, OIL, oxidation is a loss. Rig, reduction is a gain of electrons. That's what we compare those to. I need to stop playing with the yardstick. Anyways, if there is any part of this presentation should be accessible to you on Canvas, if there's any part of this presentation or the lab manual that you feel confused about, please bring it to my attention. I have another session, 8 a.m. Wednesday, MS 104. I will be there. Bring your questions. I will be ready to answer them. That will be my last review session before the exam. But if you need any other help in between now and then, please let me know. I can hop on Zoom. I can do anything. I'm relatively flexible. I won't say that I can probably drop everything, but I will be relatively flexible for you, okay? I hope this helps. Please, I'm kind of, if I can be mano a mano. Okay, let me put this back so something. If I can be completely honest, it is very hard for me to be able to make review material. Like I would love to be able to sit here and say, okay, people have been telling me that even though they haven't been making it to my review sessions, people have been telling me that they're having a little hard time with selective versus differential media types. I could make those study materials for you, and you could use them at your own will. You don't have to come to those sessions, but I could make those resources available for you. I could present the information in new and different ways. But I haven't had a direction with what exactly people have been needing help on. I've been trying to pry it, but I don't want to overstep and I don't want to be rude. I don't want to seem like I'm trying too hard or anything like that. I just genuinely want to help you guys. I know how hard of a class this is. I know everything counts when you are trying to get towards that nursing program. Your grades, they are very impacted. Doing well in this class means so much to so many people that I want to be able to help. So please reach out. Um, Dr. B and I have been trying to push this because usually people realize when it's too late. People realize when they know that they're falling behind, or people realize after the first exam and that first grade comes out, everybody's like, oh, or after the first case study, people start kind of like talking to me a little bit more. But guys, I'm like 20 years old. I'm not scary, I promise. I don't think I am. If I give RBF, it's just because I'm tired all the time and I have eye bags now. And if I'm ever like really punctile, like not punctile, I mean, well, yeah, I guess. If I'm ever like jaded or like sharp with my movements, it's because I'm on a lot of caffeine. I can't control it, I promise. Or if I talk too much, it's because there's a lot of caffeine in my system. So please reach out, tell me what's going on. I will help you through whatever you need help with. I am a resource to be utilized. And if nobody's coming to these sessions, then that means that it's one-on-one at that point. You have an hour plus with me. If you said, oh, can we meet earlier, on Mondays? Yeah, I can totally do that. If you say, what is another thing that you could say? Well, you know what I mean. If you are here at 6 and you want to stay overtime, like you're waiting on somebody else until 8, I'm also waiting on somebody else until 8, so I'll be here. And this room is completely empty. So you are free to hound me during those windows of time where it's reasonable. Please, please, please. I like interacting with you guys. It's fun. It helps me feel like I know what I'm talking about too. So yeah. So I hope everybody enjoys their night, whether you're seeing this Monday night or not. I hope this helps a lot and get in touch. Thank you. And I shall sign off now. Okay.